Single-celled organisms like archaea and bacteria emerged in Earth's oceans over 4 billion years ago, utilizing a highly efficient, minimal blueprint. This configuration has served as the baseline for life since its inception. This model consists of a selectively permeable outer membrane enclosing a cytosol gel. Within this fluid, the cell houses its ribosomes and a single free-floating loop of circular DNA. This architectural simplicity restricts these organisms to a microscopic range, typically between 0.1 and 5.0 micrometers. Eukaryotic cells, such as those found in plants and animals, can be 100 times larger in diameter. This jump in scale is not a matter of simple growth. It requires a specific internal configuration to satisfy the laws of physics. Every cell depends on its outer plasma membrane to import nutrients and export waste. Geometry presents a severe bottleneck. As a cell's radius expands, its internal volume increases cubically. Simultaneously, the surface area of the membrane only increases quadratically, failing to keep pace with the expanding volume. As the cell grows, its metabolic demand outpaces the membrane's physical capacity to transport materials. Under this baseline model, expansion is fatal. To reach a larger scale, life had to invent a different internal architecture to solve the surface area crisis. One solution to the surface area problem is to fold the outer plasma membrane deep inward into the cell's own interior. These inward folds eventually enclose the cell's linear chromosomes, forming the double-membraned nuclear envelope. This expands into the endomembrane system, a continuous labyrinthine network stemming from the nucleus that acts as a targeted transport circuit. The first specialized zone is the rough endoplasmic reticulum, which is studded with ribosomes to manufacture proteins directly into the network. The smooth endoplasmic reticulum lacks these ribosomes, focusing instead on lipid synthesis and calcium regulation. These materials travel to the Golgi apparatus, where they are chemically modified, packaged, and sorted for delivery. The endomembrane system transforms the cell into a compartmentalized industrial processing line. Internal membranes also allow highly incompatible, volatile chemical reactions to happen safely and simultaneously inside the exact same cell. Standard protein synthesis requires the cytosol to operate at a neutral pH. We see a sharp contrast in the lysosome, a specialized vesicle packed with over 60 different hydrolytic enzymes. These digestive enzymes require extreme acidity to break down waste, conditions that would damage the rest of the cell if they were not contained within these membrane-bound organelles. Without this physical quarantine, advanced processes like intracellular recycling would result in the cell digesting itself. Operating this massive infrastructure comes at an immense energy cost, one that archaeal metabolism cannot sustain alone. Roughly 2.2 billion years ago, a large archaeal host engulfed an aerobic bacterium and ultimately failed to digest it. Under the theory of symbiogenesis, this engulfed bacterium formed a permanent partnership with its host, eventually becoming the mitochondrion. In 1981, evolutionary theorist Lynn Margulis published the framework for this transition. Mitochondria possess a distinct double membrane, an architecture separate from the host's own endomembrane system. Inside, they retain an independent circular genome, a legacy of their bacterial ancestry. They also multiply independently within the cell using binary fission, the same process used by free-living bacteria. By domesticating a bacterial power plant, the eukaryotic cell secured the massive ATP yields required to sustain its size and complexity. A 100-fold increase in size introduces physical instability. A liquid cytosol cannot maintain the shape of a large cell on its own. The cytoskeleton provides a dynamic physical architecture made of microtubules, intermediate filaments, and microfilaments. This network maintains cell polarity, resists external pressure, and locks organelles in their designated coordinates. It also acts as an internal railway system for vesicles moving between organelles and the outer membrane. This same internal structure can extend through the membrane to form cilia and flagella, allowing the cell to move. The cytoskeleton is the final component that converts a passive sac of fluid into a highly directed machine. Viewed at full scale, The eukaryotic cell functions as a hyperactive, highly synchronized factory floor, processing materials continuously without pause. We can track this coordination in a single sequence. An external signal molecule binds to a receptor on the outer membrane. This triggers the nucleus to transcribe DNA instructions into messenger RNA, which exits through a nuclear pore. Ribosomes in the rough ER read this code and translate it into a raw protein sequence. After final packaging in the Golgi, the protein is carried down a cytoskeletal track by motor proteins. The vesicle merges with the outer membrane to eject the protein, a journey fueled continuously by ATP from the mitochondria. Despite their separate evolutionary origins, every organelle is integrated into a singular survival mechanism. Single-celled organisms perfected this structural marvel over 1 billion years ago. Eukaryotes utilized their endomembrane systems to secrete structural proteins and carbohydrates into the space outside their boundaries. This created the extracellular matrix, the biological glue that allowed previously independent cells to physically adhere to one another. With specialized internal compartments already in place, these communities of cells could specialize their overall functions, forming the first tissues and organs. The solutions to the laws of scale, energy, and toxicity found within the eukaryotic cell were the prerequisite for the existence of all complex plants, animals, and human beings.